Yeah, so first of all, thanks for having me. I think I was here, not here in open space before, but I think at the early days, I see Philippe there, you know, I had the privilege to be with you guys already. So what I have been asked to do actually is probably more phrase a question as to what I think. 1 second, I just need to drop this here. If at all, CRISPR, sorry about this. CRISPR has an impact on the biotech industry, and personally I think it does, but to a certain extent at least, but probably I should give you some of the reasoning, and I think we have then later on the opportunity to discuss that. Maybe before I begin, I walk you very, very briefly through what CRISPR is. So I have no slides, yeah, so I said that's probably not necessary. The other thing I wanted to mention is I have to leave directly after this presentation. I have to catch a train to get to Hamburg for another board meeting, but anyway. So I think most of you anyway know what CRISPR is, but just to summarize it, it's a, by now probably not that new anymore, gene editing technology, which allows you to make very specific cuts, double-stranded breaks on DNA, including obviously human DNA. That's one of the reasons I'm sitting here. It was actually discovered, which I always like to mention, in an effort of basic research. So at the time, Emmanuel Charpentier was looking for small RNA molecules in group A Streptococcus. Probably could talk an hour about now why she did that, but anyway, she found a system which allowed bacteria to fight viruses. The system was known, but she identified the mechanism behind that, how it could work, and in the end we and others were in the position to harness that for translation of that technology to develop drugs. Now, when I'm saying that, or at least come up with the working hypothesis, which is probably up for debate, that I would say, you know, this technology does have a significant impact on the industry, the biotech industry, by the way, also in pharma, and I will come to that and why that is. I think we should look at this context at R&D. So how is CRISPR impacting R&D? Also including large organizations, pharma organizations, drug development itself, so also clinical development, and then last but not least, certainly commercialization and reimbursement efforts. So now with the mic in my hand, it's a little tricky here, but I'm trying to manage not to lose this. So yeah, and the objective, as I said before, of this very short keynote is actually, you know, how does it, and if at all, have an impact on the industry. So I just went through that. So one thing is always important to me. You know, everybody has in the past talked about CRISPR. I do think that CRISPR is not a poster child. CRISPR is one of many new technologies. I look at this from a perspective like gene therapies, gene editing, RNAs, mRNA, antisense, CAR-Ts for sure, microbiome probably. You know, so these are all new modalities we are looking at, and I think they did benefit or do benefit dramatically from what we have observed over the last 5 to 10 years, probably, maybe 8 years, a very significant increase in knowledge and innovation in the life science industry, but also in academia. And without that, a technology like CRISPR would not be in the position to have an impact the way it has today. One classical example I always like to give that, just flipping this here, is that, for example, sequencing costs. So we all depend on sequencing. Genetic information has dropped from about $1 million, $2 million in 2004 for a single human genome sequencing to today maybe $800–900 per genome. This results obviously into a situation where we have access to a tremendous amount of genome or genetic information at very limited cost by now, and this cost is dropping. Other elements, for example, is GWAS. Probably most of you heard about that. So genome-wide association studies, which actually now give us not just a tremendous amount of information, but actually we can pinpoint, at least in certain settings, risk factors. We understand much better the cause of genetic disease and so on and so forth. I think, not true, but a few months ago I was at Swiss Re, so one of the biggest, if not the biggest reinsurance company in the world, in Zurich, discussing GWAS analysis, what it means for that industry, so it's really interesting. But then also for sure, there are elements like, when Pharma understood that probably 5, 6, 7 years ago, that there are huge, very significant clinical trials, for example, in cardiovascular, failed, or one after the other failed, or at least didn't bring the outcome they were expecting, that they kind of had to, well, I wouldn't say reinvent the model, but it was a time where we understood that we have to go much more into personalized medicine. And the reason why that is also, and this is coinciding, we obviously had more genetic information. We understood much better that this concept of one pill fits everyone is probably in many, not in all for sure, but in many indications, not the way to go. And so all that allowed a technology like CRISPR and others for sure to find their way in the industry and also to find a way to impact the industry. When I talk about R&D very briefly, it is probably not too bold of a statement to say that each large pharma company with significant R&D organizations is using CRISPR. There are a few exceptions where they are probably infringing and try to get around it, like Boehringer. I promised them to mention it, but fact is, there's a company out there, ERS Genomics, based in Ireland, who's licensing the technology, and the data show very clearly that pretty much everyone takes a license in support of their R&D organizations. And that means you're all of a sudden in the position with this and other technologies, but in particular with CRISPR to, I would say, make another step towards the direction of target validations by using really genetically defined cell lines, primary cells, animal disease models, you name it. All that for sure with CRISPR now is much easier. High throughput screening, setup of that, if you're a little smart, you can do that in many different ways. So it does have a huge impact. Even in this setting, obviously CRISPR is not used as a tool for the development of medicines. It's just a tool for R&D. Now, the other aspect is that when you think about, I think I put this down here, sorry. The other aspect is for sure drug development. I mentioned that before. That's probably not that obvious, but I will at least try to tell you why I think that is a significant impact on drug development. I already mentioned the concept of one pill fits everyone. So when you look now at personalized medicines, obviously, one of the aspects you have, at least that's a theory, and in some certain instances it works, the treatment effects you see when you treat people, are significantly larger, number one, at least in theory. They should be larger, because if you have a targeted medicine which is fully validated, then you should see a significant effect. That already from a statistical perspective has an impact on numbers and sizes of clinical trials. The other aspect is for sure when we look at the diseases we are going after, it's not only a rare disease, but in many instances it's rare disease. And again, this translates back to smaller, significantly more focused clinical trials. And that, and I can speak about that from, I would say, first-hand experience, is for sure in certain instances, or in many instances, new to regulators, and certainly new to the pharma industry. The way we look at clinical trial design today is almost research. So obviously we have to discuss that with the regulators, but in many instances we have very, very limited regulations out there. For sure we have regulations around GMP, we have regulations around cell therapies and so on and so forth. But in many instances we have, together with the regulators, design clinical trials to understand how do we make sure that on the one hand we capture treatment effects, proper treatment effects, but on the other hand obviously we provide a relatively safe medicine. And that's something which has changed with the concept of personalized medicine / CRISPR, or new modalities, the way I call that, and others I think, has significantly changed the concept of drug development in the industry, at least in the biotech industry. Now, another aspect, and then probably I close on the opening remarks, is I'm always surprised when, not surprised, but I always have kind of a reaction when I'm discussing with people, and I see a few VCs in the room, that, biotech is innovating, but in the end, pharma is developing. This is a concept we have heard over the years, and I would like to say, I think this is changing dramatically. So when you look at the valuations we have seen, in particular, most of you know that, at the East Coast in the Boston, Cambridge area, certainly also at the West Coast, a little less so here in Europe, but I think also here the valuations got up. When you look at the cash available today to companies like Moderna, CRISPR, you name them, Bluebird, this cash allows you to do a couple of things, or a number of things, I should say. It's more than a couple. So number one is, obviously, when you look into R&D efforts, you all of a sudden, obviously, can run different programs in parallel. You're not cash constraints anymore, as we often were in this industry I would say, many years back, and still, obviously, many companies still are cash constrained. That's one aspect, which you could argue is something to do with risk mitigation. Obviously, if you have a common mode of activity or action, you might be screwed anyway. But in principle, that's a consideration. What is probably more important is that is the aspect of people. So and I can just give this example for CRISPR, but it's not only CRISPR. We have access to the very best talent in the industry, and I mean industry. Not necessarily pharma, for sure. We can get pretty much anyone we want to get from pharma if there's a match. But we also can get top-notch people out of biotech. And so what we are seeing is that, I wouldn't call this a brain drain out of pharma, but we see in parts a brain drain out of certain areas in pharma into these, I would say, still probably startups, not necessarily, but kind of, biotech companies going with new modalities, be it cell therapies or whatever, going now into clinical development or being in clinical development like Bluebird briefly, or around commercialization, registration, whatsoever. And so I think the concept, in particular with regard to these new modalities, thinking that you have innovation in biotech, but let's the big boys do it, I think this is pretty much outdated. Outdated for these kind of assets. Last but not least, the comment US-Europe. Like always, to look at that, will be very short, because I think things have changed a little bit, luckily. But we, for example, built and started CRISPR in Switzerland for various different reasons. None of us was Swiss, just to claim that, make that statement. But now all of our operations are in the US, so I would say 99% of the operations are in Boston, or in Cambridge, I should say. And there were different reasons for that. Probably the most important reason was that access to cash in a very, very dynamic environment, which we at least felt, or our VCs felt, we couldn't manage that in Europe. I think when we look at what is happening in Europe, my view is that cash is still an issue, but less so. I think there's more cash available. People, in my view, don't take it personal. I think is an issue, in particular with regard to people who understand translation, development, senior executives. Not that we don't have them here in Europe, but I think access to these people is not that easy, because many of those you find in the US, and getting people back from the US to Europe is possible. We have seen this. Some of you know that anyway, also with your own companies, but it's a challenge. And then maybe there is also a little bit of an attitude, I wouldn't call it an issue, then I would label it, but an attitude challenge maybe, actually in academia, with regard to the willingness to start to think around translation, adding value to what people have done in the lab, because, and that's the last comment I make, I think when we think about innovation and research, I think Europe was in many cases number one. Checkpoint inhibitors, CRISPR, certain aspects of CAR-T, microbiome. So you could argue that most of these initiatives, most of this early thinking comes from Europe. While, and this is something we have to look at, there are always exceptions, fantastic companies here, but most of the value still is created at the East Coast, West Coast, and we should also at one point look at China. That's it.
Oh, you can
keep it. Okay.
So I would just ask you quickly one question, and then we can take some questions from the audience. I think something, in my opinion, that is impacting a lot the biotech industry linked to CRISPR is this patent war between the. Yeah, we have to discuss it quickly. So now, in my opinion, there is something important, and I think you mentioned it with Boehringer, for example, and their practice. Something that could happen because of this problem is the evolution toward the trade secret for some company, relying more on trade secrets than a clear IP with patents. And what do you think? Do you see anything happening with this, or other technologies as well, which can be linked to it?
Well, a couple of comments. I think whenever you have technologies or inventions which constitute a very, very significant value, you have these things coming up. Now, CRISPR for sure is maybe particular complex, but I think even if, I don't know if who saw it, but yesterday the USPTO declared an interference, another interference. We all knew that for sure that would happen. But I think we are beyond the point that companies like Editas or ourselves would be left hanging out dry there. So they all have now their patent estate kind of established, and now it's about some of the details. I would say who pays royalties, when and how, whatever. Trade secrets, in my view, anyway, always important. I personally still believe in protection of intellectual property, and that's one of the reasons our company, UC Berkeley, goes down this path, battling something we believe is for the right reason. But I can only encourage people to make sure that they have proper IP supporting their inventions. Trade secrets, cool. You have significant more challenges to protect that. One example I always give: we are in a zoo in Boston. We can keep, probably with the incentive programs we have, if we want to keep, most of the senior executives, but everybody else is just zooming around because the employment situation is such that it's a huge war for talent. And with that, already you have challenges to protect your trade secrets.
Yeah. Okay. We'll take a question, yeah.
Could you help us a little bit with the following question? We were facing a situation about 2 or 3 years ago that we had a basic technology which the application was relatively clear to go into drug discovery, technology, etc. How did you develop the first business plan? What did you pitch? Did you pitch that you have a technology which is basic and we license out to all the pharma and everybody is grandmother? Or did you pitch drug development from the beginning? Can you give us a
Yeah, actually, it's a good question. First of all, and I don't know who you all are, we had a business plan. It's important, yeah. The other thing is that very clearly, we had, this is in the public domain, I can say that, we had as a first investor, Versant Ventures, it's a US-based, West Coast-based investor with a pretty significant footstep in Europe, sometimes more, sometimes less. And they're known for a model which is called Inception, but a build to buy, sorry. Build to buy, where you talk to pharma, early on, with obviously a concept, a value proposition, and you provide pharma the opportunity to buy into this value proposition, means finance or fund in parts the asset and the research to get to the first value inflection point, which is most of the time something either early clinical or at least proof of concept preclinical. And then you sell it to a pre-agreed price. So,
An option deal.
that is something like almost an option deal. Now, we talked to pretty much any or every pharma company on this planet, and everybody was super excited. It was very clear they didn't get it. And the reason why they couldn't get it at the time, it doesn't fit the pharma model when you do something coming from a technology base. So it took us actually longer to convince the investor that what they were suggesting was, to be honest, not working. It was crap. So what we did, we went back to the drawing board and said, okay, let's do this differently. And I think this is something which I can always recommend to anyone who is trying to get into this business or do the second, third, fourth biotech company. So we looked at what can CRISPR do. And we knew a few things. We knew in principle it can delete, it can add genes, it can be used externally, so ex vivo you can take stem cells, modify them, or you can use the entire system, and modify something in whatever kind of target organ, be it the liver, the brain, you name it. Or we said, goodness, we want to translate this ASAP, but we want to keep it simple. So we looked for the theoretically easiest application. That was knocking out something, always easier than knocking something in. And we wanted to be able, being kind of drug developers, to control what we were doing. So we said we need a cell outside the body we can modify, a stem cell. That's the reason, and that then we then kind of picked a number of indications. We actually came up with proper development plans around preclinical development, early clinical development, everything you do with a business plan. And by the way, this business plan doesn't stand today anymore, but it probably was pretty good to be cross-referenced for about 2 years. And then we decided to, in addition to this one investor who gave us 10 million or whatever it was in the early days, to add additional investors. And by then, by the way, the technology itself was known, so we were really in a position to say, yes, no, yes, no. So this was very nice because normally we know how it works, We knock at a lot of doors. And so, but the basis was a very thorough, I would say, thought process as to how to do it. We didn't want to have the blue sky. With CRISPR, we have blue sky in our hands. We didn't want to add blue sky shooting on top. And when you look at the three CRISPR companies today, Editas and Intellia, they went down a different path. And we were 1 year behind, and it's probably fair to say that we are by now probably 1.5, if not 2 years ahead, certainly of Intellia, and probably less with Editas. And I cross my fingers, and I really mean that they will be successful because the field needs this. But it was based on a very thorough, I would say, analysis of what is doable, what is not doable, so that we can convince VCs to put a lot of money, because it was a VC financing, a lot of money into the company where we had no data. We were 1 year old, where we had an academic collaboration, which in most cases, as you all know, is very difficult to manage. We had a few data there, but we had 150 million in the bank, and we were 5 people. So that was kind of, a very unusual situation. VCs know that. But you see this more and more, by the way, in Boston now, which I personally find tricky. But then we had to deliver on that. And I have to say we did. It took us 2 years or 2.5 years to IPO the company. I was at the time the CEO. I didn't want to IPO. But the other companies, Editas and Intellia, went IPO. Our investors were pushing. Now we at least delivered on the plan. We are now a clinical stage company. We are, I just said it to you, between 2 and 3 billion market cap. We have close to 300 people, 600 or 700 million in the bank, blah, blah, blah. So on the way to become a real biotech company.
And when you say you are in advance compared to the other CRISPR companies, you're meaning in terms of
clinical development. Clinical development. It's always, in the end, it's all about data. We have officially no data, still no clinical data, but we are in clinical development. For sure, we have an idea. The others have, well, Entelia has no IND, and Editas does have an IND, but in very, so far, complex indications where the readout will just take significantly longer.
And your data are due this year?
We will, I can say that. The first data are in an indication in hemoglobinopathies, is beta thal. There is still, this is partnered with Vertex. Unfortunately, I have to leave. I have a few comments on how to do deals and not to do deals. Anyway, so we have pretty privileged relationship with Vertex on this, is 50-50. We still decide if we show these data, but I personally believe, I'm now chairing the board, so it's not my decision, but I personally believe very likely we will see some data at ASH. Yeah.
Great. Another question in the back?
So one of the big challenges with the AMTs is the fourth hurdle. So how are we going to pay for that? So we have several examples of course we market already. So thinking that from the end, do you think there is, with any of these technologies that you talk about, there's a chance to actually tackle that and really bring costs significantly down or be quicker also?
No, yeah, yes, no, yeah. Super complex, yeah, super complex. First of all, I would like to address the question a little differently, but I totally get what you mean. First of all, I think it's a very fair question. And everybody asks, can we afford this? Do we want to afford this? Yeah. So I think that's almost a question for society to answer. The other aspect is we are always afraid of these huge numbers. We have seen this now in parts with Bluebird, who they picked a number. It's not that they have a rationale behind that number, but they picked a pretty significant number. They have a rationale, but in the end they had to pick a number. They have an issue now in the US, we all know that with Medicaid, yeah, but they get there. We see Novartis, so these are very, or Unicure, we see very significant numbers. Now, what I personally believe is that we will, and you see this already with Bluebird and others, and I think that's probably the way forward. It needs to be a success-based payment where you have a certain upfront, to be debated how high, and then based on data coming in, you have new installments of payments. Now, when you think about it, that's again a question for society, do we want this? When you think about very rare genetic disease, then it's more an ethical question as to the fact, or like compared to, is there benefit to society? Probably not, thinking now altruistically, but as such, not, or very limited. Now there are diseases, when you think about in type 2 diabetes, or let's say stick first with type 1 diabetes, autoimmune disease. Well, with RegionMed combining this with CRISPR as an example, you probably can do something very significant there with regard to even cure. And that's, I think, the point. If you can cure a disease, and then you do that with young adults, for example, and then you see their productivity until the end of their life compared to what you normally see when you treat a type 1 diabetes patient right now, then there might be a benefit. But we have to think, I think, differently about these kind of questions. But it's a very fair question, and I'm pretty sure that there are countries out there who can't afford it and don't want to afford it, and there will be challenges, no question. Yeah.
I had a question. You said in this particular field there are... I'm so sorry. I had a question that, in the beginning you said that people from the pharmacy background as well as from the biotech background in like for example, CRISPR. So what is like the difference? Like what are the people from the pharmacy background and what are the people from biotech? I am a student studying biotech, so like...
Okay, so I can probably say that. I worked for Big Pharma, so I was before this the global head anti-infective Sanofi. Before that I was at Labiotech, and then Novartis. It's tricky. I think it's not so much the individual. I think a person in pharma can work in biotech, and a biotech person can for sure work in pharma. That's not a problem. I think there are a number of challenges when you look at large organization, and it doesn't have to be a pharma company, but look at a large insurance company, for example. The way decisions are made, the way budgets are approved is obviously very different than if you work in a, I would say, smaller environments, number one. Number two is that, and I think probably colleagues of mine who have worked in pharma in senior executive positions will confirm that innovation is not necessarily incentivized in pharma. For sure, they will always say, yes, it is. But I think in many instances, you come up with a plan for the year and then you need to meet that plan. And some of that is people's management. I mean, very significantly. You do that in pharma, in biotech for sure as well. When you have 3, 4, 5, or a couple of thousand people, you have to do this as well. So I think it's more a challenge of productivity, innovation. The people I've met in pharma, for example, in my groups when I was in France, fantastic. You know, I would never say anything bad about them, and I think many of them would excel in a different environment. And there's a personality question. Some people feel more comfortable in these kind of structures, while others say, hey, I want to have a different impact. Maybe I can actually knock still at the door of the CEO and say, hey man, I think what you just told me is nonsense. A little more difficult to do that in a big pharma company. You know. You can try, you know, but it doesn't always go down too well. You know.
Another question? Yeah.
More of a production-related question. I always find that when it comes to biotech and personalized medicine, one of the key drivers for cost is also that it takes quite a long time to create one therapy for one person, basically, which often takes several weeks. What's your approach to keeping this comparatively lean?
You mean manufacturing aspects? Yeah, well, no real solution. So one thing for sure is, and that's something we do at CRISPR Therapeutics, is when you think about CAR Ts, most of the CAR T cell therapies right now are autologous, which we all, you know, is very cumbersome from a manufacturing perspective. We take an approach using the CRISPR technology to switch that, knock on wood, to allogeneic therapies. That's for sure, just from its concept. That has nothing to do with pharma and biotech, just from a concept perspective for sure. If it works, much cheaper. But in general, it is a challenge. And the other challenge is with cell therapies anyway, we all know that. We don't have enough qualified manufacturers in Europe. We don't have them in the US. That's a little bit more, but not too much. And one of the reasons is that all of our companies, us included, we build our own manufacturing, at least for key components. That's probably a different session to talk about for various reasons, because it can kill your program instantaneously. In particular, if you have different modalities, cells, AAV, vectors, you name it, it's just disaster. Yeah. I think in CRISPR, the biggest R&D component is manufacturing. I think, I don't know, we have 50, 60 people, tech ops, manufacturing, just for that, at this early stage. Yeah.
On CAR T, you were working with partners as well?
No. That was still one of my decisions, and we are very happy about this. So we are the only company totally unencumbered, values with us. We have publicly announced 3 CAR T programs, 2 in classical CD19, a little boring, I admit this, BCMA, and then it's out now CD70 for renal cancer. Yeah.
Other question?
Do you believe in vivo CRISPR?
You asked the good questions. Yes, I do. I absolutely do believe in that. But and we have evidence, good evidence, supporting that kind of belief. It is still a belief, not a knowledge. I think it is just so much more complicated. Not so much from a safety perspective, by the way. That's what people always come up with. It's a safety issue. Yes, it's significantly more challenging around safety for sure. Now we have a pharmacology issue. We need to get sufficient editing in the target tissue or cell or organ, and that's still a challenge. And that's actually what I referred to at the very beginning. Many aspects come together. It's not only CRISPR. There's AAV technology, LNPs, you name it. And to be fair, you know, I think, and that's one of the challenges Intellia has together with Regeneron. Hey, I'm sure they do a fantastic job, but it's tough. It's really tough. And most of the studies you have to for profiling and qualifications in monkeys, so non-human primates, and not an easy spot to be. If your portfolio, like for Intellia, is around that kind of aspect, not so easy. Yeah, but it will come. I don't know when, but you have seen we have done maybe deals. A very nice deal. I'm very happy about that. So the team did a fantastic job with Vertex for a preclinical asset, not even in tox. We didn't even think about tox in DMD, Duchenne muscular dystrophy, for 175 million upfront, 50 million in milestones, which are to be reached within 6 months. Very nice royalties, and the entire deal value is clearly above $1 billion for something qualifying guides. It's our deal. We just did it a few weeks ago. Yeah. So, and this tells you, back to the question, that there are companies like Vertex who firmly believe, like us, in the use of CRISPR systemically or in vivo. But it won't be easy.
What's the involvement of Vertex in this particular partnership?
This DMD we licensed to them, and then we have another indication, DM1, where we have a co-development. But we said, gives us, and we have, to be honest, I think it's the largest deal ever in this kind of environment for a preclinical asset, not even having seen talks. And that's something what we see now in the CRISPR field. The value is just skyrocketing, and the reason why that is, people say, that's not justified. For sure, it's justified, kind of. I think is that if it works, it's a total game changer, and we have indications from academic studies, not our own studies, and Vertex has this as well, that in models which are really translatable, like the dog, we see fantastic data. Yeah, and so it is a game changer, and they know if you sell that program in let's say a little later, in 2 years' time, they pay 10-20x on that, and they don't want to do that.
Mm-hmm. Yeah. One last question. Yeah.
What you mean by CRISPR in vivo is to change in a tissue-specific environment, not the whole organism, right? Because the genome of the whole organism is what the Chinese scientists did.
So in vivo CRISPR.
A girl was.
I didn't. Sorry, this is blowing a little loud.
Yeah, it's a bit loud.
Sorry.
In China, they genome modified the baby.
Yes.
This is the genome modification of the whole baby.
Yeah.
What you mean by in vivo modification?
No. That's important. Yeah. Thank you that you asked this.
Yeah.
So there are two aspects, and that's really important. So one is somatic cell modification. So I take your cells and I modify them. You have babies, you won't have babies, but you make sure you have some. They won't have these changes. Yeah, this is your cell. If you go into the germline, that's what they did in China. Then for sure you inherit the genetic modification, and that's a very different story. By the way, much more interesting than IP to talk about. I think.
Sorry.
and maybe last comment there is all the companies anyway who work with that are only using somatic cell modification. But, and I have to say this, my personal opinion is at this point pretty clear. Personally, I think we should stay away from that for quite some time, not even talking about ethics. But it will come. There's no question about this, that people will use this technology at one point for germline editing. There's no question.
It would be, in a way, it would be much better because your progenitor would also be cured of a particular disease.
Sure. I always give the example of Huntington's disease. Yeah, so it's a very tricky question, but I don't think that the technology is there. But I don't think we have the framework, the legal framework, we don't have the ethical framework, and we don't understand enough yet about this technology. And there I think it's all of a sudden very, very relevant. If you have an off-site mutation, if in a somatic cell we have this in every cell cycle we have so many off-target, so means double-threaded breaks. You manipulate the germline and you screw up and you don't even know it, you have a problem. And we don't have the technology at all supporting identifying to this extent needed the analysis of what really happens in the germline. And so personally I think it's crazy. Yeah, we can't do this, and then at one point we have to ask ourselves again as a society, do we want to do this? Yeah, because this comes with a number of very significant challenges, because it's not only about treating disease. So I bet you that organizations like Pentagon or whatever, we probably know that, have programs, because it's very well known, and this is done already in cattle, how to increase muscle mass. It's very well known which kind of pain receptors can be modified to reduce pain sensation, and so on and so forth. Yeah, so there are a number of things I personally think you really want to stay away from. Yeah.
So just checking the time, maybe one very quick question. Okay, Philip.
Very quick one. I think you're one of the very rare cases, at least in Europe, where you have a top academic, a top guy from Pharma, and one of the best VC coming together and building a very big company. Why did it work with you, and why doesn't work in other cases?
You just said it. I'm joking. No, I think first of all, don't say I'm from Pharma. I take it personally. I see you have a bio shirt, so I'm joking. Yeah, so I've enjoyed my time in particular Sanofi. No, I think you have first of all, CRISPR is special. Okay, CRISPR is something we understood early. Maybe that was a particular thing, that this is easy to use. We can kind of harness that. I think this is not that often that you have access to a technology like this at this early stage. That's number one. Number two, I do think it does help if you have someone with a drug development background behind you can talk to the VCs. Where is credibility? When they say, hey, I don't know, but he has done it before, or she, and there is something, and then for sure luck. You have to be lucky, but I do think, to be very honest, if this had been my first company, it was certainly a big benefit having been in pharma. Now, really meaning that, having been in biotech, coming out of academia, having been in drug development. Having a track record, knowing VCs, understanding, having a network, because you can't do it alone. That's the other thing. We always talk about the CEOs, okay, fine, but it's about a team. I really mean that. And when you look at what kind of team we had relatively early on, Bill now left us, Bill Lundberg from Alexion was a perfect guy for the start of such a company. Maybe later on, you need different people again. But we also were in the position, because of CRISPR, to attract very smart, intelligent, humble, some of them, people. Yeah.
Is Emmanuel Charpentier active in the company?
No, Manu doesn't do anything. No, Manu is that was for us the best founder ever.
Yeah.
She's lovely. I just saw her. She's in Berlin, and she didn't get involved. And oh my God, that's great, because in this case, she didn't care so much. We knew each other for 20 plus years, so there was a relationship of trust. Huh?
How come?
Well, we met in Rockefeller in New York. Yeah, so in 1995. Yeah. And so, but that, maybe I should say this: you have fantastic founders who understand that at one point they have to maybe step back and allow others to do it, and you have not so fantastic founders who don't understand it, and then you have the founders, a few, who really can do it.
Thank you very much.